Okay, so today we're going to continue looking at biological individuality. In the last video we saw two definitions of the organism, the genetic criterion and the immunity criterion. Today we're going to look at a third approach which has been developed by Peter Godfrey-Smith, and he approaches the problem of individuality via the problem of reproduction. So he asks basically, how do we distinguish reproduction and growth? In the case of reproduction, of course, we have the creation of a new individual, whereas in the case of growth we have the development of one single individual. So if we think about the difference between reproduction and growth, this might give us some insight into the nature of individuality, into the nature of what it is to be a biological organism. So consider a herd of buffalo. A herd of buffalo grows and it may split, producing a new herd. Now we would say that the individual buffalo in the herd are reproducing, but there isn't, as it were, herd level reproduction. It's not the herd as a whole that reproduces. So we might say that the splitting of the buffalo herd is nothing but the reproduction and movement of the individual buffalo. But then this raises a question, right? Why don't we look at the individual buffalo in the same kind of way? So of an individual buffalo, you could say, well, there's nothing more going on here than just cell reproduction and then the cells organising themselves in certain ways. And that's sort of all there is. So there isn't really reproduction at the level of the organism, it's just at the level of the cell. Now this seems like a very unnatural way to describe, for example, human reproduction, or buffalo reproduction. So the question then is, what is it that an individual buffalo has that the buffalo herd lacks that makes it so that there is a genuine reproductive process going on in the particular buffalo, but not in the buffalo herd? Well, first of all, Godfrey-Smith distinguishes three types of reproduction. First, there's what he calls scaffolded reproduction. In this case, reproduction is dependent on external machinery. For example, a standard virus, say, consists simply of a DNA sequence surrounded by a protein coat, and it reproduces itself by entering a cell and causing the cell to copy the genetic material and make new protein coats. The virus essentially hijacks the genetic machinery of the cell, and it can reproduce only given the scaffolding of the cell. So left to its own devices, it's completely inert. And similarly, you have things like genes and chromosomes. Again, these reproduce themselves, but only with the aid of the cellular machinery. Then there are simple reproducers. So think of something like a single-celled bacterium. We have a single individual cell which copies itself using its own machinery of reproduction. Now, of course, all cells are scaffolded in a certain sense in that they all rely on the appropriate external conditions and energy from the environment and so on. But that's a matter of kind of providing the right nutrients and the right temperature, etc. The actual reproductive machinery, the DNA polymerases and the ribosomes and so on, that's all internal to the cell. So in a sense, the cell has its own machinery of reproduction. So a single-celled bacterium is a simple reproducer. Finally, there are collective reproducers. These are reproducing objects that are made up of various simple reproducers. All multicellular organisms are collective reproducers, perhaps also colonies and symbiotic associations such as lichen. The distinction between reproduction and growth is particularly problematic in the case of collective reproduction. That's really where the problems lie, because obviously what's going on with a collective reproducer is that in both the cases of reproduction and growth, that's going to involve cells dividing. So you're always going to wonder if you could just sort of take the reductive view and say there's really nothing more going on here other than just cells dividing. There's just reproduction at the cellular level, and that's all. So how can we distinguish reproduction from growth in collective reproduction? Well, Godfrey-Smith suggests that there are essentially three kind of criteria that we use to evaluate whether there's genuine reproduction going on. First, bottlenecking. So in an extreme case of bottlenecking, the life cycle is reduced to a single cell. And we say that the things on each side of the bottleneck are both different organisms, both different individuals. For example, in human reproduction, the parents are each reduced to a single cell. The sperm and the egg unite to form a zygote, and then the zygote develops into the new individual. And so we can sort of use that bottleneck to distinguish the one individual from another. Bottlenecks are important because they kind of force a whole new cycle of growth and development. So if development proceeds from a single cell, then all cells produced from that cell will be clonal copies. They will have pretty much the same genome. Now, as noted in the last video, due to mosaicism, the genomes of the cells in the organism will diverge over time, but they'll still be largely the same, right? Your body developed from just one cell, and so all of the other cells that develop from that are going to have a nearly identical genome. And as noted in the last video in discussing the genetic criterion, this reduces conflict. Another important feature of bottlenecking is that a mutation in the original cell will be transmitted to all the other cells. So we can sort of use this mutation to clearly distinguish the new individual from its parent or parents. That there will be something kind of special in that new individual, in that new organism that just wasn't there in the parents, and all of the cells of the new organism will have that special thing. So bottlenecking kind of creates the opportunity for significant change in that sense. Now, of course, bottlenecks come in degrees. In humans, we have a single cell bottleneck, but in the case of aspens, when they send out the runners, that's a narrowing of the life cycle because the runner is a lot smaller than the whole tree. So it's kind of narrowed, but not to the point of being a single cell. So we have a kind of partial bottlenecking going on there. A second criterion that Godfrey-Smith uses is reproductive specialization, or the germ-soma distinction. There are germline cells which are specialized for reproduction. In mammals, the germ cells are the sperm and the egg. None of the other cells in the body can grow into a new organism. The somatic cells cannot continue to reproduce, continue into a new generation, right? They die with the organism. The somatic cells just carry out those activities that are important for survival and development. Now, the key point about a germ-soma distinction is that since somatic mutations cannot be passed on to the offspring, any somatic mutation that benefits the cell at the expense of the organism will just die with it. So suppose there's a mutation in one of my liver cells which allows it to start reproducing itself rapidly, just at the expense of all of the other cells and at the expense of the overall integration of my body. I mean, that would be a cancer, right? So it's a liver cancer. Well, due to the bottlenecking, due to the germ-soma distinction, that mutation is not going to be passed on to the next generation. And so this is the way to kind of promote cooperation between cells, just as bottlenecking does. I mean, well, actually, I suppose the mutation could be passed on if there's sort of enough time, right? I mean, if it's a mutation that will produce a cancer when you're like 50 or something, then I guess it could be. But the point is that the overall integration of the new organism will be maintained, because if the mutation is in a somatic cell, it's not just going to be passed on immediately. Honeybee colonies have a very clear germ-soma distinction. Reproduction occurs through the queen and the male drones. The female workers cannot reproduce. The queen and the males are the germline. So, a honeybee colony is a kind of reproductive unit with some parts specialized for the survival of the colony and other parts specialized for reproduction of the colony. Third criterion is overall integration, and this involves a division of labor of different parts, which nevertheless all sort of function together. So in a human, we see this with how different organs play different roles and are tightly integrated to form the whole organism. The heart, liver, kidneys, and so on all do different things, and all of them need to be working in order for you to survive. So, again, both of these properties, the germ-soma distinction and overall integration, are also a matter of degree. In honeybee colonies, they exhibit some division of labour because bees are organised into different castes and they perform different tasks, but it's not quite the complexity of something like a human body. Now notice that none of the properties we've seen here are true of a buffalo herd. When a buffalo herd grows and splits, there's not necessarily any bottlenecking. There's no germ-soma distinction because all of the buffalo can, all of the sort of individual buffalo can reproduce and go into the next, go into the sort of buffalo herd that splits off, and there's no integrated division of labour. Anyway, we have three features of collective reproduction, and these features can be present in different degrees. Different cases of collective reproduction can be scored on these features and then mapped into a space, as represented here. So in this diagram we have a three-dimensional space, and each collective is given a score of 0, half, or 1. B stands for bottlenecking, G is the germ-soma distinction, and I is the overall integration and division of labour. So on the upper right, with a score of 1 for every feature, we have humans and similar animals where there's a single cell bottleneck, there's a very strict germ-soma distinction, and the cells of each unit are organised with a significant division of labour that sustains a complex whole. On the bottom left, we've got the buffalo herd, which is a mere collective because it has none of these features. And we can put other entities into this space, as you can see. In slime moulds, there's not really any bottlenecking because, as we noted, they can live as single cells. But when they aggregate, they do develop a germ-soma distinction and they do develop some division of labour. Honeybee colonies aren't represented here, but if we consider bottlenecking, well, a new honeybee colony is initiated by a fairly large swarm of a queen and many male workers, so not really any bottleneck, but there is a very strict germ-soma distinction and there is a degree of overall complexity and integration. So I suppose we'd probably put the honeybee colonies somewhere here, say sort of on just to the right of the slime mould, perhaps. Now, the point of all of this is that organisms occupy a spectrum. Organismality or individuality, it comes in degrees. Very rarely do we find sharp distinctions in nature. There is no essential property that is necessary and sufficient for something to be an organism. We just have these sort of three criteria which an entity can have to a greater or lesser extent. So that, I think, gives us a very interesting way of sort of mapping out the living world, and it allows us, I think, to see the important distinctions between something like a buffalo herd, which is just a kind of collective of particular buffalo, versus an animal, which is more than just a collective of cells. However, there are some concerns we might have about this way of thinking about organisms. So first of all, Godfrey-Smith has identified three properties that are exhibited by organisms, but we might ask, well, why stop at three? Why not say there's a fourth criterion? So we could use, for instance, degree of genetic homogeneity as the fourth criterion. And well, we saw in the last video there are certainly some reasons to favour genetic homogeneity. A fifth criterion could then be possession of an immune system, and again, there are other proposals in the literature, so this could go on pretty much indefinitely. The worry is that Godfrey-Smith's view here is kind of arbitrary, right? There's no justification for choosing the three parameters that he does. I mean, it's understandable why he limits it to three parameters, because as you start adding more and more parameters, it becomes basically impossible to apply the concept. It just becomes far too complex. but we need some principled reason for picking out the parameters that we do. And although although each parameter does have a justification in a sense, so bottlenecking and the germ-soma distinction, and the sort of overall integration, those are important points to sort of consider. They do seem to be important features of organisms. What we really need is a principled reason for using these features rather than others, rather than the other properties we might use, such as possession of an immune system. I mean, I think this point is perhaps particularly worrying, given that Godfrey-Smith's analysis only applies to collective reproduction, so it only applies to multicellular entities. Obviously, in a simple reproducer, a single-celled organism, bottlenecking and the germ-soma distinction they're just not applicable. And so we might think that, well, maybe that would suggest that bottlenecking and the germ-soma distinction are actually not really so central to reproduction or to individuality as concepts. And that would give us some reason for worrying that the parameters that have been chosen are not as important as perhaps Godfrey-Smith suggests. A second worry about this approach is, it seems like what Godfrey-Smith is doing here is taking certain paradigm organisms, as it were. He's taking entities that are uncontroversially organisms, and he's trying to identify specific properties that they have, and then he wants to evaluate all other entities on the degree to which they possess these properties. So, we sort of take a paradigm organism, say a human and other metazoans, and then we ask, how similar are the other entities to this paradigm, right? The humans and other metazoans exhibit bottlenecking, germ-soma distinction, overall integration, etc. So how similar are other entities to this? The problem with this is that, well, what counts as the paradigm? Why should we think that there are paradigmatic organisms against which all other entities should be judged? Part of the difficulty here is that the concept of organism and individual are not clearly defined in the first place. This is why we're doing this video, after all. And so, it's sort of perhaps unclear what would count as a paradigm organism. We might say, look, right, there are just many different kinds of organisms. One kind of organism is the standard multicellular animal, like a human or a buffalo. But then plants are another kind of organism. Honeybee colonies are a different kind again. In biology, variation is the norm. There is no one type of organism that is privileged. Now, if you were to start with the assumption that plants are the paradigm organism, you'd probably get a different set of properties as being sort of central to reproduction. So, if we start with plants, then maybe the strict bottlenecking wouldn't be so important. So I guess as before, we have a problem of arbitrariness here. Why should we start with humans and other similar animals? Why should we treat those as the kind of paradigm against which others are to be judged? And a final concern, which I suppose is maybe a similar point, is just that this approach, perhaps, when we take this approach of kind of evaluating all entities in terms of whether they share features with paradigm organisms, we threaten to obscure the important differences between different kinds of organisms. We might think, well, when we consider honeybee colonies and slime molds and aspens and so on, it's just misleading to think of them in terms of how well they match some ideal that has been derived from other organisms. Okay, so now I'd like to briefly outline a somewhat different side to this whole debate, and this is the recent idea of the extended organism. So the claim here is that the boundaries of the organism can extend beyond the cellular. An organism can also consist of what we might initially have thought of as inanimate matter in the environment. The first proposal along these lines was by Richard Dawkins in his book The Extended Phenotype. So biologists draw a distinction between genotype and phenotype. The genotype is the genetics of the organism, the DNA. The phenotype is the organism's observable properties, its morphology and behaviour and so on. Now, many people think of genes as coding for particular traits. You have a gene for blue eyes, a gene for aggressive behaviour, a gene for intelligence, whatever. The genes are a kind of blueprint for the construction of the organism. They carry information that specifies how the organism should be built. Now, this is basically a myth. The only thing that genes literally code for, if anything, is sequences of amino acids, so proteins, basically. Every trait of an organism is a product of interaction between many genes and the environment. There are a whole host of different influences that are involved in creating an organism. So, the situation then is genes can be thought of as being a literal code for proteins. That is perhaps debatable. It depends on what we mean by coding and so on. But there is certainly a sense in which there seems to be a kind of genetic code, and that produces the amino acids, and so we can think of it as coding for amino acids. But certainly, if we talk about genes coding for traits, that's a fairly strained metaphor. Genes causally influence the traits of an organism, but there's not like a literal coding or blueprint going on here. But the point then is that, genes don't just influence the sort of cellular body, the cellular traits. Genes will also create modifications in the environment, such as beaver dams and termite mounds. Animals are constantly altering their environments to improve their ability to survive and reproduce. So the basic argument is then, in the same way that genes influence the development of the cellular body, they also influence what we would have thought of as the external environment. So actually we should think of the phenotype as extending out into the environment. We should think of the expression of genes. If it's reasonable to talk about genes expressing themselves in an organism's bodily traits, we should also think of them as expressing themselves in the environment. One way to think of this is in terms of adaptation. And adaptation is a trait that has been evolved by natural selection. Now, it seems it's not just the cellular body that is the bearer of adaptations. The termite's mound is just as much of an adaptation as the termite's legs. So if we think of organisms as the bearers of adaptations, then it seems we have to think of organisms as extending into the environment. Now, of course, insofar as this, Dawkins's view focuses on how genes influence both the cellular body and the environment, we might wonder whether this is perhaps just assuming the genetic criterion of identity, which, as we saw in the last video, does face some problems. And Dawkins certainly did have a very gene-centric view of biology. He promoted what's known as the gene's eye view of evolution, that all processes of natural selection can be understood in terms of selection of genes. I might talk about that in another video. But the point for now is that although perhaps the way Dawkins argues for this does assume a sort of genetic criterion, there are other ways to sort of develop this idea of the extended phenotype or extended organism. And in particular, this has been developed by Scott Turner, who focuses specifically on physiological processes. So this is the maintenance of homeostasis, a stable internal equilibrium against the sort of changing environment. So this is we can think of organisms as extended, even if we look at it from a physiological point of view. Turner has done a lot of work on macrotermes termite colonies, which comprise millions of termites living symbiotically with fungi that help with digestion. The termites essentially cultivate fungus farms, and they use the fungi to break down wood, which the termites then eat as food. The fungi are responsible for about 85% of the colony's collective metabolism. So the colony is a symbiosis between fungi and termites. And similarly, with honeybee colonies, we might think of this termite fungus colony as a kind of superorganism. But Turner suggests that if we do think of this as a superorganism, it doesn't just consist of termites and fungus. So the termites build these enormous mounds, as you can see here, these pretty huge structures. Now, none of the termites live in the mounds. They live in a nest underneath. So what's the point of the mound? Well, the mound contains an intricate network of tunnels that regulate the flow of air. About 95% of the nest air is replaced once every 20 minutes. And this ventilation is powered by the heat from the nest below and from the wind energy hitting the mound. Termites modify the mound depending on the demands of respiration in order to keep the gases, the concentration of gases in the nest constant. So in particular, high levels of CO2 need to be maintained because the fungi require this to thrive. So the structure of the mound and the passageways through it are constantly being altered so as to maintain a kind of homeostasis, right? To maintain regular temperature and regular CO2 concentrations in the nest. Turner suggests then that the fungi, the termites and the mound and the nest all comprise a single superorganism. And this is an organism that is partially constructed out of noncellular material. It's constructed out of the inanimate matter that makes the mound. The mound is literally a kind of organ system which channels physical and chemical energy and is involved in respiration and temperature control. So the physiological processes don't end at the skin, basically. Okay, so that was a brief tour of some of the views about biological individuality. One point that I do want to end with is that in the last couple of videos we've spoken primarily about organisms. But it's important to bear in mind that organisms are only one type of biological individual. I've focused on organisms because individuality is just such a huge topic, and I kind of had to simplify it and find something to focus on. But in fact, there are many other entities that have been considered to be biological individuals: viruses, genes, prions, species. I mean, the key point here is that natural selection doesn't only act on organisms. I mean, certainly things like viruses and genes can be selected. And as we discussed in the first video, selection requires a population of individuals. So if we're going to talk about the selection of particular viruses, say, we need to consider, well, what is it that makes a virus an individual? And obviously that's going to introduce a whole load of other problems, because certainly the kind of criteria that we've used to think about the individuality of organisms is going to be very difficult to apply to the individuality of viruses and genes. But that's all for now. Thanks for watching.